Marie Curie Fellowship to work in between Polymath with David Meza-Reyes and the Medical University of Denver?
Yes.
Okay. And so you spent quite some time there, and after a couple of years, she returned to San Sebastian, and now she's a postdoc in Mauricio Prato's group in Biomagune. One more note, she just told me, and I found it very, very interesting, that she's been carrying with her during her entire career the project that she will be talking about today from place to place. And if I'm not mistaken, you started a startup, or you're about to, or is it secret and I'm giving out too much information?
We are starting to think about starting a startup.
Okay.
So.
Okay. So I didn't say anything about startup. So.
But I will comment it.
Yeah. So once again, thank you for coming here. I see a lot of faces which I don't see usually at the DAPC, which I think is a very good sign. And Nuria, the floor is yours.
Thank you. If you don't mind, I will talk from downstairs because I'm a small person and I'm not used to about people. So as I told, thank you, Aitzol, for inviting me here, and thank you all for wanting to listen to me. I talk a lot sometimes, and I talk about different things. So if you don't understand anything, just stop me. It doesn't have to be a monologue by myself if you don't want to. So as I told you, I'm a postdoctoral researcher in thick Biomagune. Actually, okay, we have the first problem here. Bueno.
No, it doesn't work.
No, it doesn't work. I didn't want to try it before. No.
And from your computer, otherwise?
No. Okay, I will sit here. Oh, I think it's not my problem. Something happened to me. No. Ah, yes.
No, okay.
Okay. So I am a postdoc senior in the group of Mauricio Prato in Biomagune. We are one group, but in two labs. We are placed in Trieste in Italy, and a second lab he opened here in Biomagune, where we are actually now. Here in Biomagune, we have three main research lines within the group, and I am the postdoc responsible on one of these three research lines that involve all of these kind of keywords. But to summarize it to yourself, we basically do 3D porous scaffolds or hydrogels scaffolds or any kind of scaffolds involving carbon nanotubes for neural and cardiac tissue engineering or regeneration or reconnection, or there is a lot of synonyms we can add here. So we work mainly with carbon nanotubes for this kind of biomedical applications. Well, a little bit of my past, I already told you something about that. My project is very multidisciplinary, and this is because I am a multidisciplinary scientist. Actually, I started my PhD in computational chemistry in Tarragona under the supervision of Professor Josep Maria Poblet. I studied there fullerenes, which are this kind of footballs and soccer balls, actually, but I didn't want to stick only to computational. So there I had the opportunity of performing experimental experiments actually related to my PhD. So I moved to El Paso, Denver, in the middle of nowhere, which is at the University of Texas at El Paso, with Professor Luis Echegoyen. After my PhD, well, I wanted to do more with my life. I wanted to do biomedical applications because I love biomedicine. So searching for literature, I found that the only person in the world that I liked that did fullerenes applied to biomedical science was Professor Prato. So I wrote him, hey, do you want me in your lab for a postdoc? And he was like, very suspicious. Why is a theoretical chemist coming to an organic lab? Do you have something wrong with computational chemistry? But no, it's just because I wanted to expand my knowledge. So he accepted me in 2015. And now I can say, well, I would like to think that he's happy to have me with him because we have been working very close since that very moment in Trieste. Then he opened the lab in 2016 in Biomagune, and he asked me to move here to help him settle the lab with another senior postdoc and to establish the scaffold team here in San Sebastián. This is the time when I started my collaboration with David Mecerreyes at Polymath. So we were working together, and in 2018, I started my Marie Curie fellowship between his group in Polymath and one of the collaborators of Prato in the UC Denver, Luisa Mestroni. She was a cardiologist. I wanted to learn cardiology at that time because we were doing nanotubes, scaffolds for neural tissue engineering, but we were always waiting for the biologists to test our scaffolds. So I get tired of waiting, and I told the boss, okay, Prof, I want to do biology myself. He was like, okay, so you can go with Luisa, learn cardiology there. So I did. I came back with Polymath. But when I came back, I had the problem. I was doing biology for 2 years. I have to say, at the beginning for a chemist, it was very difficult to be surrounded by only biologists. It was very difficult. It was shocking, I have to say, but I fall in love with cells. So I came back, Polymath. David has no idea of cells, no interest in having a cell culture lab in her in Polymath. So I found myself inside Biodonostia, and I asked them, okay, can you let me do my experiments here with cells? And they accepted me. They say, okay, you know cardiology, we don't know cardiology, but we are interested in learning about cardiology. And you don't know a lot of biology, so it's a good match. So since then, I am half part-time working in Biodonostia too. And after my Marie Curie, I came back to my boss, to Professor Prato, and now I'm currently working in Biomagune with tissue regeneration or cardiac. In Biodonostia and in Biomagune, we specifically do central nervous system work, also with animals. So I am here to talk to you about our project, but our project is a dream. We dream in curing spinal cord injuries, and it's a dream we share, the boss and I, we share it for a very long time. Well, you know, spinal cord injuries are injuries that are produced in the spinal cord, of course. Most of them are traumatic. Every year, half million people suffer from this kind of pathology, let's say. And in their lifetime, they have an economic burden of around $3 million per person, per patient. That some of that money they have to spend by themselves. So this kind of injury, it does not only shows paraplegia or tetraplegia to the patients, but they also suffer from chronic pain. They have physical and social barriers that are better nowadays, but there are still some of them. And for me, one of the worst things is that the probability to die prematurely, it's 5 times increase for these kind of patients. So we have to find a solution to that. This is what we want to do. There are nowadays some of the therapies, more recent therapies, for instance, we have robotics that are very advanced. And this one actually can be bought by the patients, but it's quite costly. Another kind of therapy I want to talk to you about is the implantation of microelectrodes for electrical stimulation. This guy is Brian, and in the UCLA, they implanted in their spinal cord site injury a microelectrode, and they were stimulating it for 8 months. So after this time, the researchers observed that Brian was able to move a little bit their arm. He had a complete tetraplegia. This case is important. I'm talking to you about this because conductivity is a very important asset when dealing with spinal cord injuries. And this is one of the key points of our talk and of our research, conductivity. Well, all this project, our dream, everything. No, before to saying that, sorry, this was a spoiler, but so one of the main problems that we have with the spinal cord is that it cannot regenerate as a central nervous system. So there is no antidote. We have to imagine that the spinal cord is like a cable, and when we cut that cable, there is no connection and the cable is useless. So our hypothesis is, if we can restore the conductivity, we can connect the cable again, we would be able to recover the functions that we lost due to that trauma. So that's why we want to use carbon nanotubes implants to reconnect that cable, to reconnect the spinal cord and recover the lost function. So this is our hypothesis. This hypothesis, well, again, I am confused with my presentation, it looks like. Well, carbon nanomaterials are a huge family of nanomaterials. Actually, in our group, we work with all of them, but the 3D ones, the diamond and the graphite, which is the ones that we all know about. But I'm going to focus on carbon nanotubes. These carbon nanotubes, we can understand them as roll-up graphene layers. They are cylinders, and they have very high conductivity. They are flexible, and of course, they have this cylindrical shape. These guys have been discovered in 1990, so this is not on fashion in the carbon nanomaterials world, but they are very useful. For instance, we can buy them in Sigma Aldrich, actually, with not a very high price. Here we can see a TEM of a carbon nanotube, a multi-wall carbon nanotube, meaning that we have several walls, and here we have the emptiness of the inside of our tube. This is similar what we are looking. We can see them as a bundle of randomized systems. You can see that they have some flexibility, so they are not completely rigid structures, but also scientists, not us, but other scientists grow them in this kind of beautiful forest. Well, so this idea started with the boss many years ago with one of his collaborators, which is Laura Ballerini, which is a neurologist. They were both working in Trieste. The boss was working with carbon nanomaterials, and Laura was working with neurons and trying to understand the action of the neurons. So they met together and they say, okay, why don't we mix neurons with nanotubes and see what happens? We didn't have the idea of reconnection at the beginning. They just wanted to try it as scientists. And actually, this was a great idea. In their first works, they just created a bed of nanotubes, and they incubated the cells on top of them. This is a neuron cell. This is the body of the cells, and all these elongations that you observe are like the arms of the neurons, which is where the neurons connect between each other. What Laura and the boss observed is that the nanotubes were pinching the membrane of the cells. Here we can see a nanotube. This is the cell axon, and you can see that the neuron is pinching that membrane, is penetrating the membrane. And the neuron didn't care. I mean, he didn't get angry about it. He lived happily with it. Even more, they were checking the synaptic current, which is the function of that neuronal cells, and they observed here, you have a small plot about it. They observed that in presence of nanotube, the spontaneous synaptic activity, which are these bands here, increase amazingly compared to the control, meaning no nanotubes bed. And after several works, they concluded that this effect is not only due to the conductivity of nanotube, but it also because of the cylindrical shape that the nanotubes have. So this material seems to be very promising in any kind of neuronal application. So we keep working on that. Now, in this second experiment, what they did is in the same bed of nanotubes, they put a spinal cord explant, and they check what happened. Here you can see the control image of it and the carbon nanotube image of it. You can see that there's a lot of heads, a lot of arms, hairs, exactly, sorry, with the nanotubes, that it's not as dense as in the, it's not as dense in the control, meaning that also the spinal cord explant has a feeling to the nanotubes. This for me is one of the most beautiful experiments that they did. Everything of this was before I came to the group. That's why I fall in love with the boss. So now they had these meshes again of nanotubes, and they put 2 explants of the spinal cord injury, of the spinal cord there. They were separated more than 300 microns. That means that they cannot connect spontaneously or naturally between them. So they observed that after 2 weeks, the IV, it stays in vitro. So after 2 weeks of incubation, the control, meaning the 2 explants on the glass substrate, they generated these parallel and very elongated axons. They were parallel. It's a very beautiful picture, but it's not functional, because parallel means that the lines will never cross. And if they don't cross, they cannot connect. And if they cannot connect, we are doing nothing here. But With the nanotubes, we see that there is a bunch of connections, randomized, a huge density of connections between the two spinal cords. What does it mean? Now they put two cables. I would like to say that, but they were like cables to each one of the implants and record which was the signal that explants were releasing. So they do the spontaneous behavior of the two explants and the electrical stimulation experiments. Here in this kind of graph, you can see the upper one is the left explant, this one, and the down plot is the right one. So in the control, spontaneously, you can see that none of the beatings, well, it's not a beating, none of the activity is homogeneous. But here you can see that how the two explants are synchronized. That means that they can connect between each other, they can talk between each other. So we are gaining a functionality. We are reconnecting the explants that are completely separated. Of course, with the stimulation, it works alike. We don't have any synchronization for the control experiment, but in the case of the nanotubes, you can see that both signals are completely synchronized. So this gives us value to our idea. This is the origin of our idea. Okay, let's reconnect the spinal cord. Let's do implants made of carbon nanotubes that are able to put back the conductivity that we lost, so we can recover the function. So the idea is always the same. The problem was that as the moment that I started with the box, everything that was done was in 2D. Most of the biology, 99% of the biology is done in 2D cultures, in cell plates. Everything is prepared for that. But we are not made of 2D. If we want to solve a real case, we need to be as realistic as possible. So we need to work in 3D, in 3 dimensions. So we need the scaffolds. We need a 3D substrate that is porous, it's permeable to allow the cells penetrate inside and the flow of nutrients. We need it to be mechanically similar to the real tissue, and of course we need to be it to be conductive, because again we are working with conductive cells. So this is when I started in the group, but we need to do a 3D structure substrate starting from nanotubes. Nanotubes are a powder. They are this kind of black powder that you can do nothing at the beginning with them. What we can do is we can disperse it. I mean, these are very simple ideas, but actually is what makes us succeed. So this dispersion, we are not a material scientists, we are able to control this dispersion with the functionalization of these nanotubes. We can add functionalization to them and convert them to whatever we want them to be, or at least we are trying to. So what we do is a very simple thing. We mix the nanotubes with a polymer that at the beginning is a liquid, and we create a scaffold. We use these polymers to make them build the 3D structure. So the first thing that was done was with the PDMS as scaffolds. So we mix PDMS, which is a silicone. It's a liquid that you can cure in the oven for half an hour, more or less, and you obtain a very rigid structure like rubber structure, and we were able to obtain this kind of porous scaffolds. Laura did the experiment on the explants again, the same experiment as before, and again you can see that when the nanotubes are present, the two explants are completely synchronized, as they are not without the nanotubes. So it worked. In a step further, Laura implanted this kind of scaffolds into the rat cortex to check the immunoresponse of the in vivo experiment. And after 2 weeks, he didn't observe any kind of inflammation, or the inflammation was minimum. So also worked in vivo. This was one of the first experiments that was done in implanting carbon nanotubes into a brain, actually. So we had these scaffolds that they were porous, they were mechanically stable, but which is the main problem? PDMS, it's a silicone, it's an insulator. And we had a lot of PDMS in these first various scaffolds. Even though they are kind of permeable, PDMS is bioinert. That means that the cells doesn't attach to it. They are repelled by PDMS. And even though nanotubes are conductive, PDMS is not conductive. So the conductivity of the whole system is existing, but it's not great. So in order to improve it, to do more beautiful and more functional scaffolds, we started the collaboration with David Reyes, and we decided to, instead of PDMS, use conductive polymers. These conductive, mainly polypyrrole and PDOT. These conductive polymers are very biocompatible and usually are used as coating and to create thin films of any kind of substrate, but not for creating 3D. So our goal here was to do a 3D structure using conductive polymers, which are a film, and nanotubes that are a powder. So let's see if we succeeded. What we did this first strategy. We generate a template made of nanotubes, and we use the sucrose as the porogen to generate the porosity of our final scaffolds. We did a chemical polymerization inside our scaffold, and after washing, we did obtain a 3D structure that was only composed of carbon nanotubes and conductive polymers. So we succeeded in doing this 3D from a 2D and a 1D material. Actually, it looks like a Cheeto, the chips that you eat, but in black. They have a very similar shape and, well, shape.
Yeah, how big are these things? So these are macroscopic, the scaffolds, or?
You can see, well, this one, it's 1 centimeter, but since we do the template as we want, we can make them as big as we want.
Okay.
So we can do a table of Cheetos if you want. And more or less they are, I mean, they are black because nanotubes are black. So even though this is a very beautiful project, everything in my life is black. But that's why I use so many colors in my presentations. The thing is that we were able to tune to adjust the amount of nanotubes and obtain scaffolds between 20 and 70% of nanotubes and the rest of the conductive polymer. And why is that? Why can we be able to obtain a 3D structure from a powder and a film? I mean, how is it possible? Well, this is a lot of thinking, but here are some images of the TEM images, transmission electron microscopy. Here you can see, for instance, the layer of polypyrrole in this case, and here you can see a nanotube. So the thing here is that the polymer acts as a sticker. So it sticks the nanotubes, and remember that we have the sucrose. So we have the sugar with the nanotubes around it. Then we have a sticker that is sticking the nanotubes. So once we remove the sugar, like the sticker is like it's actually the scaffold of our system. So this is why it worked. We did the micro-CT to analyze the internal structure of our Cheetos, black Cheetos, and we can see that we have 60% of porosity of our system, meaning that only 40% of it is matter, and the rest is nothing. So it's a space for the cells to grow, for the nutrients to flow, for everything to happen in this space, but in 3D. Here are some SEM images, scanning electron microscopy. We can see that we have a very homogeneous structure here too, and here a more high detail picture of the layer of polypyrrole. And here you can see like this brush, which are the nanotubes around it. Actually, we have a pore size of mainly between 50 and 100 microns. This is very important. I'm talking about porosity. It's important to have emptiness, but it's important that this emptiness is big enough to allow cells to flow inside. If imagine that a neuron is around 10 microns. If we have nanopores, even though we are nanomaterial scientists, if we have nanopores, cells don't care. Cells like they cannot penetrate, they cannot go inside. So we are doing nothing there. They will stick on the surface, and we don't want that because we want a whole 3D brick, hybrid brick between cells and material. So these are very nice results. Also, we analyze the conductivity. I told you that we did conductivity all the time. So we create this kind of 3D system to analyze the conductivity. This was challenging because at that time, there's nothing in literature that we're doing 3D conductivity. Everything that was done was in two-dimensional films. But using this technique, we observed that here we have our polypyrrole with nanotube scaffold, and we have an impedance, which is the inverse of the conductivity, that shows us that we have a system that is around 10 times more conductive than the initial PDMS. And not only that, our system is more conductive than the system without nanotubes, only polypyrrole. And polypyrrole is a superconductor. So we are having a very huge and very beautiful and high conductivity in our system. The final asset that is important in a 3D scaffold is the mechanical properties, the Young modulus. So we analyze that too. And we observe that we have Young modulus. We can change the Young modulus depending on the amount of nanotubes and polypyrrole that we have there, the ratio more than the amount. And we are moving in values within the brain system, the central nervous system, which is something that is essential for us. So we obtain a scaffold that has all the properties that we need it to be. Now the next question is, will cells like it? So we did this experiment. We introduced cells, we incubate cells inside them. Here in green, you can see living cells. They are actually cells that are alive inside our scaffold. We have a lot of them sticking around on the surface of our scaffold. Here in gray, you can see the scaffold. I told you our scaffold is black, but here it's gray. Then we have the cells here, and the black part, it's the emptiness, it's the pore. So we don't have cells here. We have cells sticked on our scaffold. Here's a more detailed image of the structure of the cells. We're using this antibody F-actin. And we also did the viability test. And this was really impressive for us. Here, remember the PDMS scaffolds. We use it as a control, and the scaffolds with polypyrrole, they more or less have the same viability, meaning that the number of cells that stick to them after 2 days is more or less the same. But look how many, this is 2.5 more times higher the value with the polypyrrole and the nanotubes, meaning that we have much more amount of cells sticking on our implants than in the previous one. So if the first PDMS scaffolds were good, these ones, they must be great. In a long-term incubation, we can see that. We have an image of that. Here you can see the PDMS scaffolds after 6 days. Okay, we have cells attached to it, it worked, okay. But look with the polypyrrole. The cells are covering the whole surface of our material. So come on, this has to be great. Our scaffold is very cell attractive, we can say. But there is also not a bad thing here, but under the chemical point of view or the fabrication point of view, this is a chemical synthesis. We need an oxidant. So we need to get rid of the oxidant because cells and oxidants, they don't match. So we need like 1 week or 2 weeks for period to get rid of all of our oxidant to clean our scaffold. So we studied a new strategy to obtain these very similar scaffolds, but with a more, I'm not going to say greener, but at least faster technology. And this is mainly done by Antonio, which was my first PhD, unofficial PhD student. We share it with David Mecer Reyes, and he designed this kind of strategy using electrochemical polymerization. At that time, electrochemical polymerization was only used for coating 2D films. Nobody used this technique to create 3D structures. But Antonio succeeded. Well, and me too, because I was also working with him hand-to-hand every day. But I will give most of the credit to him. So we generate again. In this case, I'm going to talk to you about the CNT scaffolds. And we also had a huge amount of nanotubes in our system. They were very similar cheaters as before. But in this case, I would like to point out this. Here we have a SEM image of the only period scaffold without nanotubes. And here we have the images of the period with the nanotubes. The first thing that you can see is that the thickness of our systems is half when we don't have nanotubes. And this is not because our starting template was different. No, we start from the very same templates, the very same thickness. So why is that? Here you can see from the upper part that we have this kind of holes. And for the case of with nanotubes, all these holes are kind of closed. Well, our theory, here come the material scientists, because we like to think, why do we have this thing? It doesn't happen in biology, but still in material sciences, we want to understand everything. So this was our hypothesis. Imagine that we are generating our template with sugar. These are the sugar grades. And we are placing it on top of ITO. In the case of nanotubes, all the nanotubes are surrounding the sugar. So when the electropolymerization starts, we have an increment of potential that goes from the ITO to the system. So the pidot start depositing on the conductive working electrode, which in this case is only pidot, but it's like fulfilling the holes within the first layers of sugar. But here, our conductive electrode is a 3D electrode because we have nanotubes all over the surfaces of the sugar. So we have conductivity all over the 3D dimensions of our system. So once we perform the polymerization and we wash, we did observe that the thickness of our systems is much higher because of that, because nanotubes act as a nucleation points for the pidot itself. So the polymerization takes place not only on the ITO, but all over the nanotube surface as well, because it's like a sticker, as I showed you before. Well, this is the last, most chemical thing I'm going to talk to you about today. So we use, again, cells with this, but now we use a different kind of cells. Now we use neuroblastoma cells, or as we call them, sushi cells. So we incubate these scaffolds with the sushi cells. You can see here with pidot, without pidot. You can see that after 7 days, the surface is completely covered by our sushi cells. Here are some images of more detail of the structure. But I think it's more beautiful, this SEM image that Antonio took. Here you can see that the cells inside the pidot nanotube scaffolds, they have this elongation, they have these arms typical of the neurons, but they don't see like that when the nanotubes are not present in our systems. I have to tell you at this point that these sushi cells, they are neuron to B cells. They are not still neurons, but we can differentiate them into neurons. For doing that, biologically, we need a specific protocol, specific drugs to make them mature and make them earn this arm, these axons, this kind of phenotype. What we did was not that. We just left the cells. We were material scientists trying to play with the sushi cells, say, okay, let's leave them for 2 weeks and see what happened. And what happened is that they differentiate spontaneously without doing anything else. How do we know that? Well, in biology, when you have a differentiated cell or a specific cell, you have a specific markers, like a digital print that tells you this cell is that type of cells. With neurons, we have beta-3 tubulin, which is one of the specific markers of neurons. And I painted beta-3 tubulin here in red. So the more red you see, the more neuron-like cells you have. Here at day 7, you can see that we have a lot of red here, but we don't have that much a lot of red without the nanotubes, and also at day 14. Of course, this is the graph representing this data. Of course, during the second week, the amount of tubulin decreased. And you will say, oh, but if you have differentiated the cells, yeah, but we didn't have the drug. So maybe the cells return to their original shape, non-neuron shape. But well, at least we can say that without any factor of differentiation, we were able to differentiate without willing it, because we were not looking for it, but it happened. So these are very, very promising results for implantation, for our final purpose. So we have a cell reponer here too. During this time that we perform these 3D scaffolds, because we have a dream, but I mean, we don't want to be the only ones working in this dream. So the boss still collaborates with a lot of Italian groups. So there was these guys in Rome that were constructing this kind of carbon fiber sponges. They were not carbon nanotubes as we do them, but they are carbon fibers. And they constructed this kind of spongy 3D scaffolds, only composed of these carbon fibers. So Laura used these ones to implant inside the first lumbar vertebra, which is this one here of a rat. She generated this injury. She cut a piece of the spinal cord, and he inserted the scaffold inside there. and also a control without nanotubes. And after 6 months, she saw a kind of recovery. But what I want to show you are these images taken from the MRI. These images are taken from Pedro Ramos, also a PI in Biomagune, and one worldwide known expert in MRI. And this was a very beautiful work he did. So here you can see the three visions of the spinal cord from the cut, from this part, and from the lateral and from the top. So here, this is the no injury. You can see the typical butterfly shape of the spinal cord. Here is the one that has been injured, but without nanotubes. So you see nothing, no green here, so not working. And gray is usually bad in MRI images or in this kind of images. And here you can see the black, which is the scaffold that Laura inserted. This is after 6 months. And here you can see that within the black, we can see some of the green. But here, without the nanotube, well, without the carbon material, we see nothing. In this image is, this image is representing the isotropy of the spinal cord. That means that the spinal cord is a longitudinal cable, neuronal cable. So it needs to be longitudinal, it needs to be grown in one direction to reconnect, to have a connection. So here we can see that some of these cables, some of these neurites are growing within our scaffold, even though they are not growing along the longitudinal direction that we ideally want. We did observe that even though the rats were able to recover their functions 4 times faster than the rats without this carbon material. Okay, this is a tricky thing I have to tell you. Rats and mice do recover their function of the spinal cord after an injury spontaneously. Humans, we don't, but they do. But these rats will recover with this kind of material. They recover the functions much faster than without it. So it gives us a lot of hope to our idea. So where are we now? Well, here in the lab, we still work with material science. We generate different scaffolds. Currently, we are focused on different kinds of hydrogels. We do the cell studies. So we did everything that I showed you. The new focus now is the preclinical evaluation. I'm becoming a surgeon now. So I am starting to do lesions on the spinal cord in the thoracic level, which is this one, which generates a paraplegia to the rats, and to test our scaffolds, because we have a lot of scaffolds and I want them to be tested. And the idea is, after that, with the material that we succeed, we will go to clinical trials. And hopefully, this is what Aitzol told us, that if we are lucky, we would like to create a startup of this project. Actually, we have obtained this past year, well, 3 months ago, a grant from the Foral de Guipúzcoa, and that considered as a strategic research line for the strategic plan. So we are happy to say that, but I don't write it. I'm just telling you that. Why? I mean, carbon nanotubes are in the market. I'm becoming more the startup part now. Are in the market for a long time. Actually, the carbon nanotube market is more than $6.5 billion, and 10% of this is related to healthcare, diagnosis, therapeutics, analysis, whatever. And we expect, since there is nothing, actually, there is no medical device containing nanotubes in the market up to date, we expect, due to all the scientific increase, that between 20 and 30% is going to be related to neurosciences. Again, thinking in our future startup mainly, we can say that, well, the spinal cord trauma is increasing every year, and also the market around it. So we have an opportunity here. We have a market. We have a technology that may work with very beautiful results, and we have a market we can fit in. So let's make the puzzle and see if it's going to work in the near future. So stop talking about neurons. at a certain point during all the neuron path, we thought, what about cardiomyocytes? I mean, neurons are conductive cells, cardiomyocytes are conductive cells. Neurons have calcium channels, cardiomyocytes too. So there's a lot of things that can relate neurons to cardiomyocytes. So the first day I told to a cardiologist, oh, neurons and cardiomyocytes are the similar cells. They told me, don't say that, but since you are not mainly a biologic audience, I can say that under the chemical point of view, under my view, they are more or less related. And also with the nanotubes are related, I can say. So El Capo, as we like to tell him, he let me start this collaboration with Luisa Mestroni in the UC Denver. But before I went there, they were doing these experiments. They start over again. So they had a bed of nanotubes. They incubated cardiomyocytes on their top and see what happened. In these images, they observed that the nanotubes also pinched the membrane of the cardiomyocytes, and the cardiomyocytes were okay with that. And also they observed that the connections between the cardiomyocytes increased when the nanotubes were present. And these connections are called gap junctions. And here in these images, the gap junctions are in red, colored in red. But okay, let's go to the basics. The heart is the engine of the body. We all know that. We cannot live without a heart. We can live without a brain, but not without a heart. Well, without a brain, even though it's inside it's inside our body. But the brain can be dead, the heart cannot be dead. So the heart pumps. And the and what makes it pumps are the cells that are placed inside this wall of the heart, the myocardium, that is called. This is an adult cardiomyocyte cell extracted from I think it's a rat. You can see that it has a very elongated shape and a striated pattern, and this is what it's saying us, this cardiomyocyte, it's a mature cardiomyocyte, it can beat, it has a function. This is what we are looking for in tissue engineering. And this is a cardiomyocyte, well, a cardiac, let's say cardiac tissue engineered that we did in Biodonostia. I did this kind of tissue that you can see it's spontaneously beating. It's what is more amazing about cardiomyocytes is that you know that they are working because you just place them in the small microscope that we have in the cell lab, and we just look at them, and if they are beating, they are working and they are feeling happy. So this is essential and very easy to work with, actually. So what we did in Denver is we mixed our conductive polymers and nanotubes materials with the cardiomyocytes. And we obtained videos. Well, the first time I saw this video, I was laughing alone in the lab because it was kind of funny. You can see how the engagement between the cells and the material is so close and it's so strong that the cardiomyocytes beat and it moves the material with it, and the material does not release from it. Actually, it likes it. Vale. Okay. Vale. Well, you cannot see this. I don't know why this GIF doesn't work always, but these are beating cardiomyocyte cells. This is a calcium imaging. What we do to visualize this beating is immunostaining with calcium chelate. And we are able to image the amount of calcium as it enters inside our cell, and meaning that the higher amount of calcium, the beating of its cell. And we can see here that in presence of polyviral nanotubes, the beating rhythm of these cardiomyocytes is higher than the beating rhythm without them. More in detail about this.
Did you say immunostaining?
Yes.
So how do you keep the cells alive?
It's a fluo 4 AM immunostaining.
Immunostaining is basically
No, but it's a staining, but without fixing the cells. So you have your cells alive.
You didn't use any antibodies, so it's no immunostaining, no?
No. It's a chelate. It's a chemical chelate
Okay.
that penetrates the cells.
It's a calcium chelator, yes.
Exactly. Well, I call everything immunostaining. Everything that I see in the fluorescent microscopy, I call it. Maybe it's
I
not.
was confused.
You know better than me, Carlos. So here we have some of the spontaneous plots. Here we have the control, polypyrrole period without nanotubes, and polypyrrole period with nanotubes. At first glance, you can see that without the nanotubes, the rhythm of the beating is kind of arrhythmogenic. It's not very well homogenized. But with nanotubes, it is very homogeneous and continuously beating. So one thing that we analyze with this kind of plots is the amplitude, the intensity of these beatings. And here I'm showing you the result of that. We can see that mainly none of the materials have a different amplitude, but in the case of polypyrrole nanotubes, we see a very small amplitude compared to the other ones. I have a theory about that it's not yet, I have not yet demonstrated, but my theory is that here the beats are so fast, and you will see in the next slide, are so fast that the cells do not have enough time to relax completely. So they start a new beating before finishing the previous one. So maybe that's why you have this kind of amplitude. So the next thing that we analyze in this kind of plots is the frequency of our beatings. And you can see here is the impact of our results. We can see that all the materials that have conductivity have a period that is much lower than the control one. This means that the beats per minute, the frequency of that beating is much higher. You can see here again the nanotubes with the polypyrrole presents a higher value here. Also, another thing that I would like to point out is that the error bars in any of the experiments that we do when the material has no nanotubes is higher than in the presence of nanotubes. And this is because of the arrhythmogenic behavior of the cells that I explained you before. So here we analyze the gap junctions, the connection between the cardiomyocytes when we're incubated on top of our materials. You can see here the red spots is these gap junctions that I told you about. And in the presence of nanotubes, these gap junctions are more homogenized. They follow this line, for instance, and here in the control, they are more randomized, which is not good for a mature culture. Also, I don't think you can see it properly, but all of these cells are completely striated, which is a sign of maturity. So our idea with that, okay, this was the first idea in Denver. Let's mix it and see what happens. Now that what happens is good, let's see what we are doing with that. So the idea here is to create a patch for the heart and to treat different kind of myocardium diseases. So this is our original idea. So with David, we came out with this kind of hydrogels because we need a stretchable material, and nanotubes with pyrrole or nanotubes with PIL. It's a Cheeto, it's not stretchable, it's not elastic. So with David, we were analyzing this PVA PDOT to make it conductive hydrogels. We obtained these thermoreversible hydrogels. That is good because we can shape them as we want. Actually, we can do 3D print with them in any shape that we imagine. They are stretchable and they are elastic, and they have a young modulus similar to the heart tissue. Oh, now it works. So what we did with that? Oh, I didn't. Aitzol told me to ask you about it, but I'm going to show animal images here. It's not gonna be too much. It's not gonna. I don't want to make you suffer too much, but if you have any sensitivities, just don't look at it. But if you already have, it's not that bad. So we implanted one of these patches that we did with David into a heart mice. What we wanted to test in this experiment is if there is any immune response to the animal against our material. So these animals are healthy. They don't suffer any kind of cardiomyopathy or infarct or anything. So we place it here. You can see that as soon as we place them, the patch sticks to the heart and it beats with the heart and it doesn't detach. We did also MRI. This one was done with in collaboration with Jesus Ruiz Cabello, another expert of cardiology MRI working in Biomagune. Here you can see the heart before the surgery, the heart after the surgery. I only see that the heart is working and it looks like they are more or less the same, which is a good sign, is what we are looking for. We analyze the electrocardiograms of the animal before and after the surgery, and they look more or less the same, which is also good. Also, different values I'm going to show you. I'm just going to show you this QRS value, which is something that the cardiologists refer to when talking to cardiomyopathies. This work, I have a mentor for this work, which is Ramon Querejeta, which is the chief of cardiology in the hospital, because we don't know that much about cardiology, but having his input is really interesting. And he said, okay, this patch is doing nothing to our systems. I showed him also a bunch of variables, well, not variables, but values that we extract from the different work functioning of the heart. And Querejeta also told us, yeah, everything looks nice, everything looks fine. So you have a material that it's not harmful from your body. Actually, when we sacrifice the animal, we can see that the scaffold was sticking still to the heart, which was functioning during all this time. And we also did this histology, and here we have the scaffold, and here we have the myocardium. So we can see that it's quite sticking, and we are analyzing also this data because we don't know why, but it looks like some cells have been penetrated inside our scaffold. So this is something that we have to analyze. We are actually trying to understand with new experiments. So having said all of that, I have to say now thank you to a lot of people. I call them my professional engine, actually. First of all, of course, David Mecerreyes for the opportunity of the collaboration and being a mentor and still being my mentor in sometimes. And of course, the boss, El Capo, Mauricio Prato, all our group, especially to Antonio, where is Nerea? Nerea, that is here, and Baha, which are the two non-official PhD students that I have now, and all the group, and of course, Luisa Mestroni, for teaching me everything I know about cardiology, and of course Biodonostia, Adolfo and Ainara for getting me the opportunity to keep working in Biodonostia and letting me do whatever I want, and of course Ramon Querejeta and Alejandro Elua, which is a neurosurgeon that is willing to start doing this kind of implantations to humans with me. So let's see if that goes to a good place. And thank you all for just not falling asleep and listening to me. Thank you.
Okay, well, thank you very much, Nuria. It was super interesting. So you're looking at the clock and saying, woo, but I didn't want to cut you because I was super engaged. I hope the rest were also enjoying the talk. So if you have questions, we can use some time. If anyone is in a rush, the door is there, you can use it, but I am not, and I don't think Nuria, are you in a rush? No? Okay. For those who are online, if you have questions, you have a Q&A section in the Zoom. You can use it. I think I can promote you to panelists so that you can speak, but I'm not absolutely sure, but nevertheless, use the Q&A. Is there any question in the audience? No? 1, 2. Carol, go ahead.
I don't know much about the technology and so on, but is it true that if the nanocarbons are like in the lung, there can be several problems, right? And could they possibly go into the blood and then into the lungs? Is there any information about that?
Okay, you're talking about the toxicology of the nanotubes inside the human body. Okay, I know that everybody in the world that is not in the nanocarbon world is afraid of that. it is true that nanotubes as a dust, as a powder, if you inhale them, they can create you asbestosis. They can go to your heart. They act as nanoneedles. So they are really dangerous if you don't control them. That's why the scaffolds we are doing, I didn't talk about it, but the scaffolds we are doing are non-biodegradable. So we know where they are at every time in their lifetime, which is going to be forever. We are not doing biodegradable scaffolds to lose the position or the control over the nanotubes. So we are fixing them in a place. We need them forever, because we are not going to regenerate the spinal cord or the heart. We cannot cut a piece of heart and put new cells there, because this is not how hearts work. So the applications that we have, we need them to be there forever. And this forever, as we showed in the in vivo, is not toxic. I mean, carbon nanotubes are good when you know how to use them. And the way to use them is to make them stick with your biological tissue forever in a place without moving. So there is no risk of suddenly release, go to the blood, go to your heart, go to your lungs. So we get rid of all of that problematics.
Thank you.
Carlos, you have...
So you've been showing quite a lot of images of cytoskeleton. So basically, when you're showing like actin and you're showing microtubules and things like that, and both actin, microtubules, and cytoskeleton in general are very, very important for the action, for the activity of both neurons and myocardiocytes. And in particular, for example, for myocardiocytes, the orientation of the actin is key for them, the function. And you show that the presence of the microtubules can sort of like stick inside the cells, but then
When you say microtubule, you say nanotubes?
Sorry, yeah, nanotubes.
Oh, yeah.
Carbon nanotubes, yes. So they can sort of like stick inside the cell. My question is, because I couldn't see it very much, is if those invaginations or those like affect whatsoever
Alter
the carving, yeah, the cytoskeleton of the myocardial, and then you can sort of like follow from there to the point of the microtubules. When you say that the tubulin 3, that well, I can ask you later this one. Yes.
Well, the nanotubes pinch the membrane,
Yes.
but it's like a small pinch. They do not penetrate inside the cell. So this is for first. So they do not alter the phenotype of your cell. They don't alter the actin.
They don't answer the actin whatsoever, because the actin actually is really important for the
Exactly.
for the tension of the membrane.
Exactly. So it doesn't. It's just a small pinching.
How deep
It's
is
not
that?
a penetration.
How deep, do you know how deep is that?
Why is that?
How deep into the membrane, and how?
I think it's few nanometers. Look, if we go to the very first, I didn't study this because this is something Prato and Laura did, but you can see here very well. I mean, this is the cell membrane. This is the pinching of the nanotube. It's not it. Well, I don't have here the scale, probably because I deleted it, but it's not more than, I don't think it's gonna be more than 5 nanometers, which is nothing for a cell.
All right.
No, I was saying that maybe for an axon, it's maybe for the cell body, it's not that far. I don't know either.
And then I'm a bit puzzled about the thing that you said about differentiation and then re-normalization. I'm not really a big expert in that, but what I understand is that once that you have a differentiated cell, that's it, that doesn't go back to normality. So what I'm wondering is if you're actually, so you're increasing the tubulin 3, but are you actually forming microtubules with it? Is it forming the actual structures that you should be forming? Are you actually differentiating them?
This is a question that I asked to the neuroscientists in Biodonostia, because I'm not as expert as they are. And when I talked to Pachi, which is one of the experts there, he told, because I was thinking the same of you, once you have a differentiated cell, it cannot go back, right? And he was like, well, it can. Because if you don't give them, if you are not maturing that cell 100%, a sushi cell can go back. Actually, they are trying, one of the research line that they have, and we have in common, is to differentiate sushi or iPS cells into neurons. And we can see that we need a lot of drugs, a specific medium, and at least 2 weeks to differentiate them. So when I showed them this result, they were not surprised. I mean, we are inducing the differentiation, but we are not differentiating 100% all the cells. It is possible, at least for the sushi to go back, also for the iPS. Or maybe it's just because they were dying. I mean, we didn't get into much detail on that experiment, because sushi cells are one kind of cells that we prefer to work with iPS cells now. So it's there, but it's not essentially in our work now. We just use them to test viability because they are fast and they are easy to work with.
We have one last question in the audience, and I have another online question. Well, Luca, you have another question too. Well,
Thank you.
I'll
start. Okay, I have a question for the chemistry and one physical question. The chemical one is the PSS. You showed Peter PSS, which is a fantastic material. If you use PSS, which is a very strong acid, you can dope it. You can even get, if you do it right, metallic conductivity in the polymers, which is incredible. But on the other hand, the PSS, polystyrene sulfonic acid, is not just some acid, it's a strong acid. So whenever we thought of doing some conductive polymers, I told my people, no, don't try it. Exactly. Of course, they should do it. As I see now, how do the cells survive that? I mean, this is like putting HCl, concentrated HCl in the medium.
No, because it's not in an acidic form, it's neutralized. I mean,
well, it's doping the P dot, but it's not using all the protons.
But when you put the media there, the media has a pH of 5.5. So even though the PSS, it's solvating the P dot, it's doping the P dot, there is a counterbalance there. You have a lot of media there.
So you undope your P dot in the
yeah, more.
So you're losing.
This is one of the main problems that we have with P dot PSS is that they detach. So that's why in our P dot nanotubes, we don't use PSS. We are using the nanotubes as a dopant for the P dot. Yes.
And more general question, but it's a very complicated for me at least. I understand you use electrodes in the human body, like in the brain or in the heart, to stimulate something. I can, as an electrochemist, somehow I have some idea how that could work. What I don't understand is if you put a conductive material in it. There is no conductive material in nature, as far as I know. Maybe melanin, I don't know. But between cells, how do the cells communicate? The cells communicate normally electrochemically, not by electricity, not by electrons. Does anybody know how that works and why it could work? We see that it does.
Yeah, this is the question.
Okay.
We have been dealing with Laura, with the neuroscientists, with Prato and myself. We are like trying to understand how this can work. Why nanotubes what are they doing? They are getting the neurotransmitters and making them move faster. They are moving electrons. What is happening? We don't understand that. And we don't know which experiment would do that. I was thinking, I don't know, a cryo-TEM with a specific marker for the electrons, which is something that doesn't exist. So we don't really know that. But if anybody has an idea of an experiment to answer this question, we will be really, really happy. But we don't know why, but we know that it worked.
Well,
just to follow up this question. So if you use, for example, purely semiconductor, the carbon tubes, could you solve this problem? I mean, because I expect here you use multi-wall carbon nanotubes, which are metallic. But if you use somehow semiconducting.
Well, these are semiconductive nanotubes.
All of them, really?
So they are not.
But they should be single
they are
wall to be.
Okay. They are called metallic nanotubes, but they are not as conductive as PDOT, for instance, in the best way of PDOT. So really, I mean, under behind the curtain, they are considered in electrochemistry, they are considered semiconductive. in biology or in material science, we are considering them conductive.
I was thinking about something a bit different when we distinguish between carbon nanotubes, purely semiconducting and metallic.
Yes.
So having one and the other, you could somehow say what happens with the lazy connection between neurons.
But that is a very nice idea. We didn't try it.
But to have but then you will need to go to the single wall carbon nanotubes, which is very difficult to work with, and so on.
Yeah. We were trying. I mean, the first experiments that we are doing, whenever we are trying to do nanotubes with anything, from mainly organic chemistry, we use single or even double wall carbon nanotubes. But it's crazy working with them because they cannot disperse and they are so in that material that it's yeah. Yeah, but trying to use a multi-wall carbon nanotubes that have a different known conductivity, even less, it's not a bad idea.
There's lots of material,
Oh, we can cover them with something.
but intrinsically they'll be metallic because they have many nanotubes inside, multi-wall.
Yeah.
Okay. Well, Luca, you have
Just a curiosity.
the last question here.
Okay, just a curiosity. And it's related to a question they just asked. Is there anything related to this type of work with metals, metal conductivity? And how is it performing compared to these systems?
Well, Laura and the boss, actually, this is why I told you here that the cylinder and the conductivity, it's important here. They did experiments with cylindrical non-conductive materials, nanomaterials, and with conductive materials as graphene, similar works, and they didn't work as good as nanotubes.
Because I'm totally ignorant in the field, what could be the reason if no,
We don't know. We only know that the shape and the conductivity, this is the big question I was telling him. I mean, we don't know how it works physically or chemically or electrochemically, how it works. We only know that nanotubes are like a shortcut for the neurons. This is the hypothesis that we have. We don't know exactly what is moving there, but we know that it works. This is something that we want to answer, but we don't have, at least we don't have the ideas to know how to answer it.
Usually they use ions, right, in between cells.
Yes, but what is happening here? We are moving the ions 300 microns.
Maybe they are like nano pipes.
Another thing, I didn't show you that, but there's another very beautiful experiment that it's a video actually that Laura did. And imagine that Aitzol is one neuron, I am another neuron, and we don't see each other. But within nanotubes within us, we are able to find each other. And there's this video that shows how the two neurons grow axons until they find each other. And without the nanotubes, the axons are just not growing towards one another.
So that's another possibility.
Yeah.
Okay, I had some problems with the Q&A online, but I read a question, which is an excellent question to finish. Somebody in the audience asked in Spanish, actually, but I hope he or she is fine with us talking in English. A very generalist question, which is the $1 million question. So when are we going to see this applied to humans? Because there are some people who are already requesting applications like this. That's
well, actually, before Christmas, I was talking to this doctor in the Goodman Institute, which is an institute of rehabilitation for spinal cord injury patients. And when I was explaining to him my idea, well, our idea, he was like, oh, if you give me material, we can start next year or in 2 years, the clinical trials. We can do clinical and we can do preclinical at the same time. And I was like, whoa, wait. So if you ask him, next month. If you ask me, 3 years. And if you ask the technology transfer of Biomagune, 10 years. So it depending on who you ask.
but it's approaching. That's
yes, I hope.
If you ask me about my research, I would tell you 50 years, 100 years or something like that, because I have no idea of when it's going to happen. So
We expect it to be,
that's
to be soon.
very good news. Okay, so with this, I think we can close the session. Please join me thanking Nuria for this amazing talk. Qué guay. ¿Qué?
Si estaban incluidos, no.
Estamos fatal. No tenía sentido, ¿no? Nos fuimos a demasiado. Me enseñó los planos, el problema es eso, me enseñó demasiadas cosas. No, me ha dado mucha ilusión. Lo que pasa es que vuestra idea, por ejemplo,
No ha cambiado.
coger una planta de hacer laboratorios en otros lados es imposible. Porque los arquitectos les han puesto como condición que todos los laboratorios tienen que ir en el mismo momento que
Sí.
Ay, qué tonto. Hay que estar todos cubiertos.
Pero en el TEM no. No. O sea, cuando tú ves el TEM.
Vamos fuera, es mejor. Estamos fuera, ¿vale?
Vale.
Venga.
Que le enseño una cosa y salgo.
Entonces.
Mira, cuando yo pensé, claro, nosotros pensábamos en que el jefe, perdón, el jefe me decía, oye, pero esto los estás cubriendo todos y tal. Pero no, mira, o sea, tú tienes aquí, estos son nanotubos, tú tienes nanotubos sueltos. Es como, es un punto de nucleación, pero luego el polímero crece donde él quiere y engancha los nanotubos alrededor. No, todos no.
Claro,
Una parte que está recubierta, obviamente, porque se tienen que enganchar.
es un proceso muy rápido.
Sí. Claro, es que nos dura.
Es que yo en la tesis trabajé.
La primerización hecha en
Yo
3D.
en la tesis trabajé en el recubrimiento de nanotubos de carbono con sílica, ¿no? En disolución.
Sí,
Todos estaban bien recubiertos con
claro,
sílica.
pero porque los querías
Sí,
solubles,
todo bien.
¿no?
Sí, todo bien. Sí, por eso me llamó un poco la atención, porque pensaba que, claro, que si fuese denso, lo que podría provocar que todos están recubiertos, dejan de ser nanotubos, porque es un polímero.
Exacto, pero no, porque además piensa que también los tengo enganchados con el azúcar por una parte, entonces todos esos que están enganchados con el azúcar, que luego le quito, están libres. No sé qué tengo que hacer,
Esa
madre.
es otra pregunta, o sea, ¿cómo hacéis los puros? O sea, el azúcar, ¿cómo lo metéis?
Nosotros hacemos una pastita, le metemos al azúcar un poquitillo de agua, muy poca, 10 microlitros, y lo mezclamos con nanotubos. Entonces hacemos como esta pasta negra. Entonces el azúcar no se disuelve del todo, solo la capa de fuera.
¿Tenéis algún tipo de control sobre el tamaño de
Sí.
de las pistas de azúcar?
Sí, es muy handmade. Hemos comprado unos filtros así que...
¿Podéis controlar el tamaño del poro?
Sí, porque tú ya compras, no sé cómo se llaman unas mallas porosas, lo pongas para escurrir la pasta. Pero castellano. Pues eso. Pues una es de 100 micras y el otro es de 250. Entonces ya le compramos eso a Sigma y metemos ahí la azúcar y entonces tenemos solamente el tamaño.
He cogido todo, pero va a quedar el close. ¿Del otro lado? ¿Sale esta parte? No, esta se va. Las... Sí, pero claro, pues solo alguna. Yo hago todos mis y hago lo que
Ayer un poco mejor.